Cell division by Sam Evans and Ginny Hoyt. Mitosis, important for somatic cell division and asexual reproduction. Cells spend most of their time in interphase. This phase consists of G1, S phase, where DNA is replicated, and G2, where the cell prepares for division. This is a picture of G2. Chromosomes are still decondensed, and at this point, spindle microtubules start to organize around the centrosomes.
In prophase, the chromatin becomes tightly coiled and packed into chromosomes that are easier to separate than loose chromatin is. Asters start to form out of the centrosomes, lining up microtubules in preparation of later steps. The mitotic spindle begins to form and the nucleolus disappears. In prometaphase, both kinetochore and non-kinetochore microtubules form, with kinetochore microtubules beginning to attach to some kinetochores, starting to line them up for metaphase. The nuclear envelope has dissolved by this point, so the chromosomes can start to move to the metaphase plate. In metaphase, the chromosomes are lined up at the metaphase plate, with two kinetochore microtubules attached to each chromosome, one on each sister chromatid. The two centrosomes are at each pole of the cell, organizing microtubules. By this point, the non-kinetochore microtubules have stretched across the cell, preparing to start cell elongation. The chromosomes that were just lined up at the metaphase plate now separate and are pulled by the kinetochore microtubules away from the metaphase plate. These kinetochore microtubules are disassembled by a motor protein on the chromosome end of the microtubule. The non-kinetochore microtubules have started their work elongating the cell in the direction of the poles. Anaphase is the shortest phase in mitosis. Telophase refers to the decondensing of chromatin, the reforming of the nuclear membrane and nucleolus, and how each daughter cell returns to regular function. Cytokinesis refers to the splitting of the cytoplasm and occurs concurrently with telophase. Cytokinesis in animals involves a cleavage furrow forming and involves a cell plate forming in plants. Mitosis creates two genetically identical daughter cells with the same number of chromosomes as the parent cell.
Meiosis. Prophase 1. Chromosomes begin to condense and homologs loosely pair along their lengths aligned gene by gene. The paired homologs become physically connected to each other by a zipper-like protein called the synaptonemal complex. This state is called synapsis. During synapsis, crossing over occurs and non-sister chromatids exchange segments of DNA molecules. Synapsis ends and disassembles the synaptonemal complex, which moves the chromosomes slightly apart. The homologous chromosomes have an X-shaped region called the chiasmata, which exists at the point where crossing over occurs. It is a cross because the sister chromatid cohesion still exists throughout the crossing. There is centrosome movement and spindle formation. The nuclear envelope begins to break down. Near the end of prophase, microtubules from either pole begin to connect to the kinetochores. These homologous pairs then begin to move towards the metaphase plate.
In metaphase 1, the pairs of homologous chromosomes are now aligned along the metaphase plate with one chromosome in each pair facing a pole. Microtubules from either pole touch the kinetochores of either homolog. Since the chromosomes line up on the metaphase plate independently, that creates the law of independent assortment, which increases genetic variation in a population.
Anaphase 1. Homologous chromosomes separate and are pulled in by microtubules towards opposite poles.
In telophase 1 in cytokinesis, each cell has a set of duplicated haploid chromosomes. Each chromosome is composed of two sister chromatids, and at least one of the sister chromatids contains a segment of non-sister DNA. Cytokinesis occurs simultaneously with telophase and divides the cytoplasm, forming two daughter haploid cells. In animal cells, a cleavage furrow forms. In plant cells, a cell plate forms. No chromosome duplication occurs between the next step due to the presence of sister chromatids.
Prophase 2. Spindle apparatus forms and sister chromatids attached at the centromere gravitate towards the metaphase plate. Metaphase 2. The chromosomes are positioned at the metaphase plate. Due to crossing over, the attached sister chromatids are not identical. The kinetochores of the sister chromatids are attached to microtubules extended from the poles. Anaphase 2. Sister chromatids separate and microtubules pull them in towards opposite poles. Telophase 2 and cytokinesis. Nuclei form and chromosomes begin to decondense. Cytokinesis is occurring.
The end product of both stages of meiosis is 4 genetically distinct daughter cells, each with half the chromosome number of the parent cell. The main difference between meiosis 1 and 2, is that in meiosis I, homologous pairs separate, and in meiosis II, sister chromatids separate at the metaphase plate.
Mitosis results in 2 identical diploid cells, while meiosis results in 4 genetically distinct haploid cells.